Okay, stuff that you need to know about living organisms consists of cells. First point, Schleiden and Swan. Theory developed in 1840s, their cell theory, and it basically said three things. Number one, all living things consist of cells. Number two, new cells are formed only by the division of pre-existing cells. And last point, that the cell contains information that acts as the instruction for growth, so chromosomes that you see here. So microscopes are pretty basic if you're looking at light microscopes, and there are certain things that you have to remember about them. So you have to remember that up here you've got your eyepiece lenses here and you've got your objective lenses here. To work out the magnification, you multiply the eyepiece magnification by the objective lens magnification to give you a result. So if this was, say, times 10 up here and this was times 10 down here, then you'd end up with times 100. Simple, really easy. But there are two key things you have to remember when you're talking about microscopes. One of those is about magnification. Make sure that you're able to quote the definition for that. So it's the degree to which the size of the image is larger than the object itself. And the second is resolution. And here you're talking about the amount of detail that you can see. So think about it in terms of the number of pixels that you have on the screen. So the more pixels you have, the greater the resolution, the greater detail you can see. Figures that you have to wearisomely remember is that magnification for a light microscope is times 1,500, and resolution for a light microscope is 200 nanometers. So that's as good as you can see. And I can't write with this. Okay, really wibbly wobbly stuff like bits of brain. So if you remember me telling you, if you put someone's brain in your hand, it would actually just fall through your fingers because it's so wibbly wobbly. If you want to make a slide out of that, you have to embed it into wax and then section it. So that's what you can see they're doing here. And then the last thing you need to know about is staining. So if you don't stain a sample, you pretty much can't see anything because most things are colourless in cells. So we have to stain them to make different bits of the cell show up. And there's two stains that you need to remember about, and they are acetic orsein, which is probably not said like that, but it's brilliant for staining DNA chromosomes, and so used so that you can study the process of mitosis when you're using a light microscope. And gentian violet, which is used to stain bacterial cell walls.